Hello class, today we are going to learn about the T cells and cellular immunity. T cells are the basis of cellular immunity, also called cell-mediated immunity. Cellular immunity depends on T cells to eliminate intracellular pathogens, reject foreign tissue recognized as non-self, and destroy tumor cells. T cells do not directly bind to antigen. Instead, phagocytic cells such as macrophages or dendritic cells process and present antigenic peptide to them. T cells have T cell receptor, TCRs, that recognize an antigenic peptide attached to a specialized presenting molecule on the cell. When T cells are activated, some destroy the target cells that present a particular antigenic peptide. Others can proliferate and secrete chemical messengers called cytokines and induce other cells to perform a function. APCs associated with cellular immunity include B cells, dendritic cells, and activated macrophages. All APCs have MHCs on their surfaces that present potential antigen fragments to T cells. T cells interacting with the antigenic epitope and the MHC will lead to T cell activation. APCs produce IL-12, which activates Th1 cells, which are activated by antigen present by MHC class II. After binding to antigen presented by APC, CD4 cells secrete cytokines activating other T cells and B cells. Th1 cells activate cells involved in cellular immunity. Th2 stimulates the production of eosinophiles, IgM and IgE, which activated by endogenous antigen and MHC class I. When activated, transform into CTLs and memory cells. CTLs lyse target cells or induce apoptosis. MHC class I on all nucleated cells, MHC class II on surface of APCs. T helper cells are classified as CD4+, which bind to MHC class II molecules on B cells and other APCs. CTL cells are classified as CD8+, which bind to MHC class I molecules. The CTL recognizes endogenous antigens on the target cell's surface that are in combination with an MHC class I molecule. A CTL attaches to the target cell and releases a pore-forming protein, perforin. Pore formation contributes to the subsequent death of the cell and is similar to the action of the complement membrane attack complex described granzymes, proteases that induce apoptosis, then able to enter through the pore. CTLs are not only cells that can lead to the destruction of the target cell. NK cells can also destroy certain virus-infected cells and tumor cells and can attack parasites, which are normally much larger than bacteria. NK cells are large granular leukocytes. In contrast to CTLs, NK cells are not immunologically specific, meaning that they do not need to be stimulated by a particular antigen. Instead, NK cells distinguish normal cells from transformed cells or cells infected with intracellular pathogens. NK cells first contact the target cells and determine whether it expresses MHC class I self-antigens. If it does not, which is often the case in the early stage of viral infection, and with some infecting viruses that have developed a system interfering with the usual presentation of antigen on an APC, they kill the target cell by mechanisms similar to that of the CTL. Tumor cells also have an induced number of MHC class I molecules on their surfaces. NK cells cause pores to form in the target cells, which leads to either lysis or apoptosis. In antibody-dependent cell-mediated cytotoxicity, NK cells and macrophages lyse antibody-coated cells. Antibody-mediated immune responses intensify after the primary response, where a particular antigen is first met and the corresponding antibodies are produced. This secondary response is also called memory response. IgM appears first in response to the initial exposure. IgG follows and provides longer-term immunity. The second exposure to the same antigen stimulates the memory cells to rapidly reproduce a large amount of antibodies. The antibodies produced in response to this secondary exposure are mostly IgG. The goal of self-tolerant is to eliminate B and T cells and recognize self molecules. Apoptosis occurs when the clone of the B cells in bone marrow is lost. When the negative selection of the T cells in the thymus fails, it can lead to autoantibodies and autoimmune disease. Active immunity, protection via introduction of antigen into responsive host. For example, naturally acquired via infection or artificial acquired via vaccination. Passive immunity, protect via transfer of antibodies or immune cells to a non-immune host. For example, artificially acquired via vaccination injection of immune serum after exposure. Both actively acquired immunity and passively acquired immunity can be obtained by natural or artificial means. Natural active, including natural exposure to antigen-induced an immune response, and immunity following an attack of measles. Natural passive, including transfer of antibodies or cells produced by others, and a temporary immunity from antibodies of the mother transferred to the infant across the placenta or in milk. Artificial active, including deliberate exposure to antigen induces an immune response, and the immunization of children. Artificial passive, including antibodies in immune serum, are introduced into body, and injection of rabies immune globulin after a dog bite. Thank you for your attention.